Hello everyone, good evening. We'll start in just few seconds. So let's begin. So in the previous class we studied about applications of enzymes in different fields, mainly in food and food industry, which includes um, milk industry, meat industry, and fruit and wine juice industry, and alcohol industry as well. So, and we little bit also mentioned about diagnostic enzymes used in diagnostic field. So these are the different applications of of enzyme applications in different industries. So in food industry we already studied about different types of food industries, which is alcohol and wine. Yeah. So in food industry we have alcohol and wine industry. We have dairy, in which there is milk production, cheese production. Then we have baking industry, and finally we also have meat industry. So these are all part, and we also studied which all enzymes are required in these uh, uh, in these industries. And in the medical field, we last time we studied little bit about diagnostic field. So in diagnostic field there are different enzymes which could be diagnosed, and these enzymes are basically will help us to predict different organ damage or different diseases. We'll study more about medical field apart from diagnostics in today's class, and then finally we'll move on to environmental field. So these are the applications of enzymes in the medical field. So in disease diagnosis, as we already studied that uh, we can measure different organ damage or other diseases or disorders by estimating the enzyme present in the blood or directly from any tissue sample. So for example, we can measure glucose by enzyme glucose dehydrogenase or glucose oxidase. So this enzyme will essentially convert. So basically here we are not actually measuring any enzymes related to diabetes mellitus, but we are actually converting the glucose. So we take the blood sample, and then from that blood sample we estimate glucose. So we are estimating glucose here instead of any other enzyme, and this estimation is done by two enzymes, glucose oxidase and glucose dehydrogenase. So this glucose oxidase, it is, as the name suggests, oxidase. It will oxidize glucose and it will convert into gluconic acid. And then this gluconic acid can be estimated by either it could give some color in an assay, or it would give you some electrical signal, and that signal or that chromogenic compound can be directly be proportional to the glucose concentration, and thus we can estimate the glucose concentration, that is fasting glucose level and normal glucose level after meal. And thus we can check. So these tests are very widely done every day for diabetes patients to regulate their diabetes levels. So this was part of disease diagnosis. Then enzymes can be also used for treatment of different diseases. So there are different neurodegenerative diseases like Alzheimer's, Parkinson's and epilepsy. So these diseases are treated by the compound that is detected during these neurodegenerative diseases is GABA, that is gamma aminobutyric acid. So this GABA will convert pyridoxal 5-phosphate into some chemical or into some substrate that can be estimated electrically. So these GABA levels can tell us the disease, how the disease has progressed, or how the disease has gone to severity, or how it is, it will tell us basically the condition of the diseases. So the enzyme here it used is glutamate decarboxylase. So this glutamate decarboxylate basically will decarboxylize the gamma aminobutyric acid, okay, and it will convert it to a intermediate molecule which is pyridoxal 5-phosphate. Similarly, for diseases or like atherosclerosis or hypercholesterolemia. So atherosclerosis is nothing but deposition of cholesterol or fatty lipid molecules into the blood vessels, which leads to the blockage of the blood vessels and ultimately can lead to high blood pressure or heart attack. So here the compound that we detect is cholesterol, and this is detected by the enzyme cholesterol oxidase. So these are the few examples in which enzymes are used for the diagnostic purposes. So this cholesterol oxidase, again it's a oxidizing enzyme. It will convert cholesterol to cholesterol 4-ene and 3-1. Again this is a compound that can be estimated and which will be directly proportional to the cholesterol levels in the blood. Then we have urease enzyme that will break down urea, and it will convert to urea to ammonia and carbon dioxide. Then we have lactate oxidase, tyrosinase and acetylcholinesterase, as we studied in the previous class, that this acetylcholinesterase is the this enzyme is present in the it's present in the nerve endings, and it is responsible for the muscle contraction. So so this enzyme will inhibit the it will break down the acetylcholine molecules, which is the molecule for the acetylcholine receptor present on the nerves, and it's a neurotransmitter. Acetylcholine is a neurotransmitter. So this acetylcholinesterase is used for the treatment of Alzheimer's disease. So these are the different applications that it is used for the treatment of different diseases, like for acetylcholinesterase is used for the treatment for Alzheimer's, and it can be used for diagnosis. It can be used for hormone regulation. It could be used for different anti-inflammatory agents, like it could be painkillers or different anti-inflammatory agents to reduce the inflammations, or it could be a biosensor also for the estimation of different like we studied in the previous class. ELISA it's a type of a biosensor for diagnostic to measure different virus levels in the blood. So let's study each of the enzymes involved, like very roughly, which are the enzymes involved in different diseases. So one of such major disease is a cancer, which is quite prevalent these days. So cancer is it's a condition that is controlled by different factors. So we have here normal cells, which divide in a controlled manner, whereas cancerous cells, when we call cells cancerous, they divide uncontrollably. So we have normal controlled division. And how this is controlled division of cells, that is controlled multiplication of cells, and this process is controlled by homeostasis, and this homeostasis is disturbed in case of cancer. And when the homeostasis is disturbed in case of cancer, it could lead to uncontrollably division of cells, which could lead to multiplication of cells, and thus it could lead to what? Cancer or tumor formation. So there could be abnormalities in DNA repair. There could be abnormalities in cell death pathways, that is the cells, there is no homeostasis maintained because there is no programmed cell death like apoptosis happening to the cells. So the cells keep on dividing and there is no escape for them, there is no death for them. So they evade this controlled death by programmed cell death known as apoptosis. So so there could be DNA repair damage. So there are different DNA damaging agents present in the cell, and these DNA damaging agents, once the damage is encountered, if the cell is not able to fix it, the cell is supposed to die through programmed cell death, through apoptosis or other forms of programmed cell death. But if it evades that DNA damage, it can lead to genetic instability, it could lead to different mutations, and with this mutation it again can lead to, and those mutations can sometimes lead to uncontrolled growth or uncontrolled division. So evading cell death is one of the factor, and there could be some genetic instability or mutations in the tumor suppressor or tumor suppressor genes or tumor genes, which could lead to cancer again. Then another characteristic of cells is that we have telomeres at the end, and after every division these telomeres they are shrinked by every division. But in case of cancer cells, they have seen very high levels of telomerase activity, which leads to again causing them to divide rapidly, indefinitely, and form tumors. And these gene mutations, again, it could be either from DNA repair or it could be other mutagens which are present in the environment, like cyanogen bromide, or there are different mutagens which actually can mutate the genome randomly, and these mutations can lead to uncontrolled or growth of uncontrolled divisions of these cancer cells, which will ultimately lead to tumor. So here is the quick representation of cell cycle and how the cells keep on dividing by inhibiting the checkpoints. And this is the normal cell. There is division and there is healthy tissue here, and as you can see, the cell growth here is controlled, that is, there is no excess cell growth happening. Whenever there is excess cell growth, that is taken care by apoptosis or any other programmed cell death. But in case of cancer, there is some changes happening, genetic level changes, and it just divides rapidly, which could lead to a formation of a tumor or uncontrolled cell mass. So there are what are the different causes of cancer? So these are the different causes of cancer which I mentioned earlier also. So there could be environmental factors, as you can see here. So environmental factors could be exposure to carcinogens like tobacco, UV radiations, asbestos, arsenic, formaldehyde, or it could also be due to poor diet or lack of physical activity, which can lead to inflammation in the body, and which could lead to again increase in ROS, that is reactive oxygen species, and lead to DNA damage. There are different genetic factors like mutations in oncogenes, as I said, these tumor suppressor genes, and these spontaneous mutations. So these mutations are prone to cause different changes in the DNA, which can ultimately lead to cancer. Then DNA damage. So there are different ways that DNA can be damaged, like ROS, UV, different radiations, replication error. So DNA can be damaged by itself also while replicating, but that is sometimes taken care of. But when the damage is persistent, it can lead to mutation, which can ultimately lead to cancer. Uncontrolled growth and proliferation, that is dysregulation in the checkpoints, the cell cycle checkpoints, which can lead to increase in proliferation of cells, which can ultimately lead to tumor. Then different infections like many viruses like hepatitis virus, hepatitis B or C, they can also lead to cancer. So they can, these viruses basically integrate their genome into the host cell, and they genetically alter our genome at specific regions, and which can lead to cancer. And different bacterial toxins are also responsible to cause cancer. Then we have different oncogenes. So these are tumor suppressor genes, KRAS, MYC, BRAF, EGFR. So any mutations in these genes can lead to cancer. For example, this BRCA1 and 2, this gene is responsible for the breast cancer, and any mutations in this BRCA1, 2 can lead to breast cancer. Okay. So these are the different factors which can lead to cancer. So these are the different enzymes that we can target in order to control the cancer. So here comes the application part, that which all enzymes can be used to target the cancer. So as we know, there are different enzymes which are involved in the cancer. So we have alkylating agents. So these are the drug classes. So we have alkylating agents are those drugs which will alkylate the DNA. Okay, so these are the examples of drug, cyclophosphamide. So they will cross-link the DNA and prevents replication and induce apoptosis. So these drugs, cisplatin, they will basically induce apoptosis. So these drugs will target mostly the cells which are very rapidly dividing, and cancer cells are the ones which are rapidly dividing. So they will specifically, we cannot say specifically, but they are more prone to target cancer cells. But during the chemotherapy, normal cells can also undergo some cell death. That's why people are more prone to other infections, because the immunity is also weakened that time. So that targets the DNA polymerase enzymes. So as we know that cancer cells are rapidly proliferating, so there will be more DNA polymerase in action. So these enzymes will basically target the DNA pol enzyme, and it will stop the cancer proliferation. Then we have different anti-metabolite like 5-fluorouracil, methotrexate, and gemcitabine. So these disrupt DNA synthesis. They mimic the naturally occurring nucleotide, like 5-fluorouracil. It mimics cytosine. So it will get integrated into the DNA synthesis, which will ultimately lead to DNA break, and then it will lead to apoptosis. So they target thymidylate synthase, dihydrofolate reductase, So this enzyme is mostly used in purine synthesis, and they also target DNA polymerase. So again, so this thymidylate synthase and dihydrofolate reductase are used in the synthesis of purine and pyrimidines. So again, it will block the DNA replication, that is rapidly dividing cancer cells. Then we have topoisomerase inhibitor like doxorubicin and etopside. So they will inhibit the activity of topoisomerase, and these topoisomerase basically are DNA relaxing enzyme. They will relieve the tensions in the DNA strand during replication, and if these enzymes are not present, there will be a strain or tension in the DNA and the DNA will break. So ultimately that will lead to DNA breakage and thus stopping the cancer. Then we have mitotic inhibitors like paclitaxel and vincristine. So these disrupt the microtubules and prevent chromosome segregation. So if the chromosome is not properly segregated, the main target enzyme is tubulin enzyme. So if the chromosomes is not properly segregated, it will lead to the blockage in the cell cycle checkpoint and it will lead to apoptosis. Then we have DNMT inhibitors, D-cytidine, azacitidine. So this class of inhibitors are widely used for chemotherapy as a chemotherapeutic drugs. They inhibit DNA methyltransferase enzymes and add methyl groups to the DNA. So they regulate the gene expression and thus it will control the cancer cell proliferation. So there are different metabolic disorders. So what are these metabolic disorders? So metabolic disorders are nothing but which are caused by different metabolic enzymes like phenylalanine hydroxylase. So this phenylalanine hydroxylase, it will convert phenylalanine to tyrosine. So these enzymes are responsible in the metabolic pathways like amino acid, like formation of different amino acids in Krebs cycle, in glycolysis, in TCA cycle, and in lipid beta oxidation pathway. So all these enzymes are involved in those pathways in order to control the any diseases. So phenylketonuria, it's a disorder. Okay, it's a metabolic disorder, and in this disorder we need to break down this phenylalanine, because this phenylalanine is responsible for phenylketonuria. So the defective enzyme is phenylalanine hydroxylase, and what is the therapy for this? The therapy is low phenylalanine diet and gene therapy. Then next is galactosemia. So in galactosemia is nothing but galactosemia is galactose in blood. So basically body is not able to digest galactose. So people mostly prefer galactose free diet, and which could lead to galactose free diet, so they will not encounter this galactosemia. Then we have different metabolic diseases like Gaucher's disease, Tay-Sachs disease. So these are the different metabolic diseases, and these are the defective enzymes which are not able, which is not functioning, or maybe there is some problem in the genetic level, which can lead to these metabolic disorders. Then we have AIDS, that is autoimmune deficiency disorder. So in AIDS is caused by virus, HIV virus, and AIDS is it's a disorder, it's a complication of HIV virus, and these are the different enzymes which are found in the HIV virus. So first is the main reverse transcriptase enzyme. So as the name suggests, reverse transcriptase, it converts viral RNA to DNA. That is, its role is that the viral RNA which is infected, it is converted into DNA, and then that DNA is integrated into the host genome. Okay, so we have different drugs which target this reverse transcriptase. So we have different, these are the examples of the drugs. So they will inhibit the viral replication, and thus they will decrease the load of the virus. Then then we have different other enzymes like protease, integrase, and GP41. So these are the enzymes which are required during the packaging and integration processes of the viruses. So for example, proteases, they cleaves the viral polyproteins. So we have different huge proteins which are cleaved to form which are cleaved by these protease enzyme to give us different proteins which are required for viral packaging and replications. So so here we are using different class. So all these drugs have VIR at the end, which suggests that they are antiviral drugs. So, and these drugs are protease inhibitors. They will specifically target the viral protease present in the body. So these if we target these viral proteases, there won't be any viral proteins for viral replication, and it will basically block the mature viral proteins and thus viral replication. Then we have integrase enzyme. So integrase enzyme is required for the viral DNA to be integrated into the host cell during genome replication. And then we have different integrase inhibitors like as mentioned here. So they inhibit the viral replication similarly. Then we move on to diabetes. So as we mentioned earlier also and studied that there are different enzymes which are involved in diabetes. So first enzyme is alpha glucosidase. So this alpha glucosidase break down carbohydrates into glucose in small intestine. So the drugs that target this are arabose and migitol. So basically alpha glucosidase will break down more carbohydrates and it will increase the blood glucose level. So these drugs will target the alpha glucosidase enzyme directly, and it will reduce the activity of this enzyme, and thus it will decrease the blood glucose level. Similarly, we have dipeptidyl peptidase, DPP. So what is this role? It regulates insulin secretion by inactivating the incretin hormones GLP-1 and GIP. So these are the drugs that targets GLP-1 and GIP. So once we decrease the GLP-1 and GIP levels, it will promote more insulin secretion, and after promoting insulin secretion, the blood glucose level will ultimately go down. Then we have sodium glucose co-transporters, SGL2. So these glucose transporters, they basically reabsorb glucose from the blood, and they are taken up by the kidneys and they are reabsorbed from the blood, and during the urine excretion also the glucose is again taken up by the organs. So these are the so this regulates the blood glucose level again. Then we have insulin degrading enzyme, IDE. So why do we want to degrade insulin in the liver or other tissues? It is because increasing insulin level, so basically sometimes there is insulin resistance, that is how much ever blood glucose how much our blood glucose levels are increased, the insulin keeps on producing, and there is no regulation, so we need to regulate insulin also. So we have insulin degrading enzymes, and then we have glycogen phosphorylase. So this glycogen phosphorylase will break down glycogen in the liver and muscles to release glucose. So this phosphorylase activity, there are no current drug targets for this, and this activity has been studied for the diabetes treatment. Then we move on to liver cirrhosis disease. So these are the different enzymes which are targeted. Matrix metalloproteases. So they break down the extracellular matrix, and doxycycline is a drug which is widely used in research also, and it is used to target this enzyme to inhibit the MMP activity. Then we have tissue inhibitors, like tissue inhibitors of metalloproteases. So so these N-acetyl cysteine is the drug that will inhibit the tissue metalloproteinases, and they increase the TIMP1 levels, which inhibit the MMP activity indirectly. Collagen cross-linking enzymes, LOX and LOXL. So beta BAPN, beta aminopropionitrile, it promotes the collagen cross-linking, and thus it will inhibit the LOX and LOXL activity. So these are the different drugs which are used to treat liver cirrhosis by inhibiting cytochrome P50 enzymes, glutamate cysteine ligase, and alcohol dehydrogenase. So alcohol dehydrogenase and aldehyde dehydrogenase, LDH, ALDH and ADH. So these are the major enzymes which are used also in the diagnostic field to check the liver damage as well. So these are the enzymes which are responsible for the alcohol metabolism, and these enzymes, so whenever there is liver damage or fatty liver, these enzymes could be increased in the bloodstream, which would tell us that there is some liver issue, and so there are different drugs which are used to control these enzyme levels in order to metabolize alcohol. Then we move on to neurodegenerative diseases. So so we already studied acetylcholinesterase, which is used to treat Alzheimer's and Parkinson's disease. So this acetylcholinesterase, it will break down acetylcholine, and acetylcholine is a neurotransmitter which works on the different nerve endings. So the mechanism of these drugs are they reversibly inhibit the acetylcholinesterase enzyme, and it will sufficiently decrease the acetylcholine levels in the brain. Monoamine oxidase, this is another important enzyme after acetylcholinesterase. So this monoamine oxidases, they irreversibly inhibit the monoamine oxidase B, and reducing the breakdown of the dopamine in the brain. So this enzyme basically breaks down monoamine neurotransmitters to dopamine, epinephrine and serotonin, leading to different neurodegeneration disease in Parkinson's. So these are the drugs that target this monoamine oxidase. Then we have tyrosine hydroxylase, TH. So this enzyme is again involved in the Parkinson's disease. So it is involved in the production of dopamine again. So this is the drug levodopa, which treats which targets tyrosine hydroxylase in Parkinson's. Then we have catechol O methyltransferase. This enzyme is used for the breakdown of levodopa and dopamine and reducing the efficacy in Parkinson's disease. So we need to inhibit this enzyme COMT in order to increase the levodopa and dopamine availability. Then we have GABA, that is glutamine butyryl aminotransferase, which is involved in the metabolism of GABA, and deficiency of GABA can lead to increase in levels in epilepsy. So we need to inhibit GABA T levels in order to maintain the GABA levels in the brain. So if we take lot of drugs, so these drugs need to be also flushed out from the body, and there are different metabolic enzymes which are involved in flushing out of these drugs, mainly in liver. So in liver we have different liver and kidney. In order to excrete all these drugs, so we have different enzymes, cytochrome P50 and UDP glucouronyl transferase, sulfotransferases, so monoamine oxidase. So these basically enzymes are involved in the excretion or solubilization of the drugs from the body, and then flushing those drugs out from the body. So coming back to the last field of enzymes involved in the environment, so we have enzymes involved in the bioremediation. It is involved in wastewater treatment, which is very important. In biofuel production, again one of the important industry and widely growing industry, and the last is paper and pulp industry. So in bioremediation we need to break down organic pollutants. So we use n number of enzymes like lacases, peroxidases, dehalogenases, P40 monooxygenases. So the organic pollutant first need to be degraded in order to for the further treatment, and then in bioremediation we also have breakdown of oil spills. So lipases are widely used for this oil spills. And in wastewater treatment we have breakdown on fats and oil. So again lipases are used. For protein breakdown we have proteases, and for starch and glycogen breakdown we have amylases. For removing nitrogen based compounds we have nitrifying and denitrifying bacteria. For biofuel production we need to break down cellulose into lignocellulose, so we have cellulase enzymes. And then we also need to break down starch and glycogen, then again we have amylases. And production of methane from organic waste we have methanogenic bacteria. And in paper and pulp industry we again need to break down lignin, so we have lacases or lignase. And for breakdown of cellulose we have cellulases. So this is just a brief introduction. What are the cellulases? We have already studied that these enzymes, they hydrolyze the glycosidic bond present in the cellulose, and not only cellulose but lignocellulose and hemicellulose. So cellulose is a crystalline matrix of beta 1,4 D-glucose linkage. So this beta 1,4 linkage, so in starch and glycogen we have alpha 1,4 linkage, and in cellulose we have beta 1,4 linkage. That's the only difference. Hemicellulose comprises of different compounds like xylins, xyloglycans, arabinoxylins and mannose, forming complex with substitutes such as acetyl esters in its backbone. Hemicellulose and cellulose are cell wall component in the plants, where the hemicellulose forms hydrogen bonds with the cellulose and other hemicellulose, thereby providing mechanical strength and stability to the plant cell walls. So in cellulose, glucose molecules are unbranched, okay, they are completely unbranched. They are straight chain glucose molecules with a long polymeric structures. So cellulolytic organisms such as T. raceae produces three major enzymes: endogluconases, exogluconases and beta glucosidases or cellobios. So endogluconases, as the name suggests, they will chop from the between. Exogluconases, they will start degrading cellulose from the ends, either from the reducing end or from the non-reducing ends. And then we have beta glucosidases or cellobiosis. So we have different products which are trademarked by which has as the enzymes. So we have care enzyme. So they have monocomponent alkaline cellulose, and these are isolated from microorganisms like Aspergillus oryzae and Humicola insolens, and the application is in laundry and detergent production. Then we have Cellubrix. So this enzyme contains cellulose and cellobiose enzymes which are in application in fruit juice and alcohol industry. So these are the different cellulose which are present, and the enzymes which could either be as per the application. We have different types of enzymes isolated from microorganisms mostly, and there are applications in baking, fruit juice, laundry, stone washing, wine treatment. So this table consists of that. Then we move on to chitinases. So in chitinases, chitinase are group of hydrolytic enzymes that catalyze depolymerization of chitin. So chitin is present in different insects. It's a linear polymer of N-acetylglucosamine chain, and so the chitinases basically hydrolyze this N-acetylglucosamine chains, okay, and they convert it into monomers or trimers or dimers. So what is chitin? Again, chitin are found in the shells of crustaceans, exoskeletons of insects, cell walls of fungus, microfauna and planktons. Commercial sources of chitins are shrimp, crabs and lobster waste. Naturally, chitin is bound to protein, lipid, pigments, minerals like calcium carbonate, and therefore chitin preparation involves demineralization and deproteinization of chitinous waste using strong acid and base. So again, chitinases are of three types: endochitinases, chitobiase, and beta and acetylglucosamine base. So these are the different types of chitinases, and the function is different based upon chitinases. They will, like for example, endochitinases, they will randomly hydrolyze the chain of the chitin residues. So these are the different sources from where we can isolate chitinases, mostly from bacteria and fungi, and these are the applications of chitinases enzymes in different industry. Mostly they are used as a biocontrol agent, and for the generation of fungal protoplast and as a biocontrol agent. So they are also widely used in industries and pharmaceutical and clinical as well. So they are also used in the in chemical insecticides and different biocontrol industries. Next we move on to lipases. So lipases again, they will hydrolyze the triglycerol linkages, triglycerides. So the physiological roles of lipase is to hydrolyze triglyceride into di-mono fatty acids and glycerol. The types of reaction they catalyze are esterification, acidolysis, transesterification, alcoholysis, aminolysis, hydrolysis, based upon the type of enzyme, which is there are different types of lipases, so each has a specific activity. There are different groups of different types of so now we move on to pectinases. So pectin, again, it's found in the plant cell wall, and pectin it's very important to digest pectin from this plant wall, especially in juice industry or paper industry, and also in the environmental. So this pectin is hydrolyzed by pectinases, and so yeah, it is extensively used in food, juice, and wine industry. So, and it also helps in removal of inner wall of lotus cell, garlic, almond, and peanut. So these are the different pectinases which are present and the type of reaction it catalyzes. So now we move on to the assignment questions. The enzyme involved in the production of dopamine. So dopamine synthesis basically the dopamine synthesis involves tyrosine hydroxylase. So the answer is D. So we have dopamine synthesis in 3-4 step, and tyrosine dopamine is made from tyrosine amino acid. So tyrosine hydroxylase is the answer. The enzyme that converts viral RNA to DNA during viral replication cycle is, so RNA to DNA is converted by reverse transcriptase. Topoisomerase is just to release the tension in the DNA. Tubulin is again involved in the cell cycle. DNA polymerase is involved in DNA synthesis. But from RNA to DNA conversion, we have reverse transcriptase enzyme. Match the disorder with their respective defective enzymes. Fabry disease, Gaucher disease, Tay-Sachs disease, Pompe disease. So the correct order is same. Fabry disease and their defective enzyme is alpha galactosidase A. Gaucher disease defective enzyme is glucocerebrosidase. Tay-Sachs disease defective enzyme is hexosaminase A, and Pompe disease defective enzyme is acid alpha glucosidase. So the correct is A1, B2, C3 and D4. The role of catalase in the food industry. So catalase is involved in breakdown of H2O2. So it will convert H2O2 into, this we studied in the last class also, into H2O plus O2 gas. So the correct answer is decomposing of hydrogen peroxide. Breaking down of fats is done by lipases. Improving dough texture is again by cellulases. And reducing amylase, not cellulase, amylase. Reducing browning in fruits is by different enzyme. The enzyme that is commonly used in the production of bread to improve the dough elasticity and volume is, so for bread we use amylase enzyme, as I mentioned in the previous question. It will improve the dough elasticity and volume. So amylase will basically break down starch to glucose. and it will improve the dough elasticity and volume. Match the drugs with the enzyme inhibited by it. 5-fluorouracil. 5-fluorouracil So 5-fluorouracil will inhibit thymidylate synthase in the biosynthesis pathway of purines. Acyclovir will inhibit DNA polymerase. Lovastatin will inhibit HMG CoA reductase, and 6-mercaptopurine will inhibit adenyl succinate synthase. So the correct answer is A1, B3, C2D1. The primary function of endoglucanase in cellulose degradation. So endoglucanases, that is, it will randomly chop off glucose. So the primary function is to hydrolyze the internal cellulose bonds. In the paper industry or in also in the environmental field. Match the drugs with their mechanism of action. Vincristine. So vincristine will disrupt the microtubule. Then irinotecan will inhibit the activity of topoisomerase. A4B3, C2D1. Cisplatin is the it's a metal complex, so it will cross-link with DNA to prevent replication and it will induce apoptosis. So cisplatin is the cross-linking agent of DNA. Then D-cytobine, it will inhibit the DNA methyl transferase activity, and thus it will inhibit the expression of few genes. The organophosphatases, which is commonly used as irreversible acetylcholinesterase inhibitor in insecticides, is parathion. D. Answer is D. The end product of chitinase activity on chitin molecule. So the chitin molecule, chitin is a homopolymer of N-acetylglucosamine. So the end product will be N-acetylglucosamine. It's a homopolymer, that is, it is made up of long chains of N-acetylglucosamine molecules. The type of chitinase acts on internal beta-1,4 glycosidic linkage of chitin chain is. So internal internal will be always endochitin. It will chop off the chitin from in between. Exo will chop off chitin from the ends. Chitobiase, chitin synthase, no. So the correct answer is chitinase, endochitinase. The end product of triglyceride hydrolysis by lipases is fatty acids and glycerol. So we have glycerol molecule, and this is the fatty acyl chain, CH2 linked fatty acyl chain. So if the lipases will break here, so we will be having fatty acid and a glycerol molecule. In pharmaceutical industry, lipases may be employed for antibiotic production, digestive system health, drug delivery system, respiratory system. So lipases are mostly involved in the drug delivery system. The effect of pectinase treatment on fruit texture. So it increases firmness, enhance crispiness, softening, enhancing the strength. So pectinase, basically they are found in the fruit cell wall. So if we have a fruit, it is present in the fruit skin. So basically we are targeting this fruit skin so that we can get juice more easily. So basically we soften the fruit, and we just soften the fruit overall layer, and then these fruits are further pressed for juice. And the enzyme which is often elevated in blood of individuals with liver cirrhosis, indicating liver damage, is so the enzyme is ALT, that is alanine aminotransferase. So that's it. Any questions? More of the questions, ma'am, more of the questions here aren't from lecture alone. I'm pretty afraid about them. Do we need to read some books, or just lecture notes is enough? So yeah, so the lecture might not cover everything. Okay, so you have to so these questions are pretty basic, then you don't have to read anything extra. I think notes is fine. The lecture is fine, you don't have to read anything extra. Like there are few questions like which are different diagnostics. For example, this question is from the lecture itself. So most of the question will come from the lecture itself. There are just one or two tricky questions that are just which involves little bit of to understand the overall application part. You need to do little bit of, you don't have to do it, it will be just one or two questions. So the exam will be mostly MCQ based only, right? So mostly I think the exam will be MCQs based, so you don't have to worry much. I think one or two questions will be just a little bit different in the context only of the topic, but otherwise it will be fine. All the best. Do you have any other doubt? Okay, thank you. All the best.